This is TreponemaPallidum, the bacterium responsible for the sexually transmitted infection syphilis. It is a gram-negative spirochete that relies on endoflagella located between the cytoplasmic membrane and the outer membrane of the cell to propel itself throughout the host. T. pallidum is transmitted most frequently through direct contact between sexual partners who have been exposed to the primary or secondary lesions on the skin. It can also invade the host through the immune-compromised tissues such as the eye and placental membrane from the affected mother to the unborn child. Unlike other gram-negative bacteria, T. pallidum lacks a lipopolysaccharide layer, which is often used as an endotoxin that elicits an immune response in the host. Without this LPS layer, T. pallidum is able to avoid the immune system activation and continue infection. T. pallidum's outer surface is unique and only has rare integral proteins in the membrane. These proteins, also known as TROMPs, allow the bacteria to evade the adaptive immune system since antibodies are unable to bind. T. pallidum is also able to bind host lactoferrin proteins to its outer membrane and acquire iron, which will enhance its survival in the host. This is also beneficial because the bacteria cell will now have host proteins present and will not be detected by the immune system. T. pallidum is able to attach to a wide variety of cell types such as epithelial and fibroblast-like cells. Two specific TROMPs bind to fibronectin on the host cells. They are TP0155 and TP0483. These two proteins bind to both matrix-insoluble and soluble forms of fibronectin. T. pallidum also has a laminin binding protein, TP0751. Laminin is associated with the basement membrane of cell layers. These three proteins are essential for attachment, and studies have shown that attaching to host cells is inhibited when anti-fibronectin and anti-laminin antibodies are used. Binding with these two components of the extracellular matrix of host cells allows the bacterium to pass through the cell layers and inhabit the human host. How this pathogen invades the host is still not well understood. T. pallidum can pass through intercellular junctions of endothelial cell monolayers, which allows access to the bloodstream. T. pallidum can motivate cellular production of matrix metalloproteinase 1, the enzyme that breaks down collagen. This may be a strategy utilized by the bacterium to invade host cells. As mentioned previously, this bacterial species does not have an LPS layer, which means no endotoxin. Let's say this fuzzy little creature gets inside your body, evades the host defenses, and starts colonizing. What can you do next? The main antibiotic used for syphilis is penicillin, and thankfully there aren't any signs of T. pallidum gaining resistance. Azithromycin has been shown to work as long as the disease is caught early enough, in the primary stage. If left untreated, syphilis can have terrible effects on the body, including gumas that can develop in the skin, bones, and other organs of the body, and neurological complications can develop. There are currently no vaccines available for syphilis.